Welcome back from the break. We're on the home stretch now. Moving into session 3 on noteworthy guidances for injectable products. Our presenters include Dr. Xingnan Zheng, Staff Fellow, Division of Therapeutic Performance 1, Office of Research and Standards in OGD, and Dr. Eric Pang, Senior Chemist, Division of Therapeutic Performance 1 in the Office of Research and Standards in OGD. Please join me to welcome our first presenter, Dr. Zhang.
Thank you for the introduction. Today, I will share the considerations for the in vitro BE approach of injectable drug substance suspension products using medroxyprogesterone acetate as a case study. By the end of my talk, you will be able to understand how the in vitro BE approach developed for the injectable drug substance suspension, describe the product-specific guidance on medroxyprogesterone acetate, recognize the common deficiencies observed for the in vitro BE study, as well as the data and the information be submitted for the in vitro BE study in ANDA submission. So, what is injectable drug substance suspension? In injectable drug substance suspension, the particles are generally made up only of micronized or nano-sized drug substance. The drug substance particles are the only insoluble component in the formulation or reconstitute formulation. For other excipient in the formulation, they are all soluble in the solution phase. The drug release for drug substance suspension does not rely on the release controlling excipient, for example, PLJ copolymer. The drug substance suspension has a simple drug release mechanism, then suspension products contain release controlling excipient. The dissolution rate is generally determined by particle size of drug substance and the solubility of the drug substance, as described by Noyes-Whitney equation shown here. When demonstrating bioequivalence, the utilized BE study should be sensitive, accurate, and reproducible. For systemically acting injectable drug substance suspension, FDA generally recommend the in vivo BE study with PK endpoints. In addition to in vivo BE studies, FDA has also recommended in vitro BE study for some injectable drug substance suspension products. Next, I will walk you through how the in vitro BE approach developed using a case study of PSG on medroxyprogesterone acetate. First, let us check the product-specific guidance PSG on medroxyprogesterone acetate. The PSG recommends two options to demonstrate BE. Option 1 is in vitro studies, and option 2 is in vivo BE study with PK endpoint. You may choose in vivo or in vitro approach to demonstrate bioequivalence between the test and the reference products. Load per Regulation 21 CFR 314.94 Drug product for parenteral use must contain the same inactive ingredients in the same concentration as the ILD, with allowable difference with regard to preservative buffer or antioxidant, provided the difference do not affect the safety and efficacy of the proposed product. Following in vitro BE study, our general expectation is that the proposed test product is Q1, Q2 the same as the ILD to ensure the identical formulation components to begin with. We acknowledge the Q1-Q2 sameness alone may not be sufficient to ensure comparable formulation characteristics between test and ILD products, because difference may arise from manufacturing process and/or excipient source. Therefore, based on our understanding, we also recommend a list of comparative physicochemical characterization to be conducted to ensure comparable formulation characteristics. For example, polymorphic form, crystalline shape, and morphology of the API, appearance, pH, osmolarity, specific gravity, sedimentation rate and volume, and viscosity over a range of shear rate of the drug product. When a suspension product is formulated Q1, Q2 the same as an ILD, there could be difference in the arrangement of matter within the dosage form, which may impact drug product performance. For injectable drug substance suspension, it is recognized the particle size can impact drug release. This has been discussed in slide 3, described by Lois-Whitney equation. We also have GDUFA research conducted for understanding the relationship of the product critical quality attributes to the in vivo performance using Medroxyprogesterone acetate as a model drug. Formulations with identical composition but different particle size were prepared. In vitro drug release methods with discriminatory power and reproducibility were developed. An in vivo PK study was conducted in rabbit to study the relationship between the particle size and in vivo performance. A level A IVIVC was established using data obtained from USP apparatus 4 with semi-solid adapter method. Therefore, with the improved understanding in drug substance injectable suspension, as well as our internal knowledge obtained from end-application data, the in vitro approach was recommended for Medroxyprogesterone acetate injectable suspension. Then, let us see how an in vitro approach being executed in an end application. This is a high-level summary of the information and data to be included. First of all, a composition table that shows the test product is Q1, Q2 the same as the ILD should be provided. The following study should be conducted on the test product comparing to the reference standard, the IS products. Comparative physicochemical characterization. BE Study 1, particle size and particle size distribution. BE Study 2, in vitro drug release. For particle size distribution, laser diffraction is generally used. We acknowledge the particle size measurement instrument setup is based on first principle. However, per regulatory perspective, it is expected end application provide data for method development and method validation. The particle size study should also contain the statistical analysis that the particle size distribution should be compared using population bioequivalence based on D50 and span. The applicant should provide no fewer than 10 datasets from 3 different batches of both test and IS products for PBE analysis. If an applicant uses a different statistical method, it is recommended the end applicant provide sufficient justification and enough data to support the proposed statistical method. For the in vitro drug release study, similarly, it should include the method development and validation study to support the utilized in vitro release study is properly developed and validated. The equivalence should be established using a proper statistical method from at least 3 batches of tests and reference standard products. One suggested statistical method is model-independent similarity factor. The common deficiencies in PSD study is that there is no method development and/or validation report. We are highlighting the method development for particle size measurement because the flocculation associated with injectable drug substance suspension. In formulation development for drug substance suspension, stabilizer or viscosity agent may be used to induce flocculation and prevent agglomeration or outward rippling. Flocculation is a process by which small primary particles in suspension collide to form loosely held larger secondary particles called flocculates. The polarized light microscopy here shows what it looks like. The smaller particles are 1 to 2 micron and are primary particles. The larger flocculated secondary particles are around 10 microns. The interconversion between these two types of particles is reversible and highly dependent on the shear condition. As shown in the figure over here, flocculation could introduce variations in particle size. Therefore, if the method is not well developed and validated, the variations in particle size measurement due to flocculation could impact the PBE conclusion. Here is a summary of things to consider for particle size distribution study. For method development, how the study analysis parameters have been chosen should be included. For example, how the dispersant, sample dilution range, stirring speed selected for measurement. It is also recommended to include precision and robustness study for method validation. For the particle size result report, it should contain full profiles of the particle size distribution, particle size result, as well as the statistical analysis. Next are the common deficiencies for the IVRT study. One common deficiency observed in the IVRT study is that there is no method development study. It was observed some end applicants use the FDA method from the dissolution methods database without additional information and rationale to support the IVRT method is suitable for assessing the proposed generic product. Another common deficiency is that there is no validation report. It is expected end applicant provide data to support the utilized IVRT method can discriminate the critical quality attributes of the drug product, for example, particle size. In addition to lack of method development and validation study, we also observe lack of statistical analysis of the release data in the ANDA submission. Therefore, for IVRT study, method development report should be provided to include how the experimental parameters are selected. For example, how the media composition, volume, stirring rate be selected. The IVRT study should also be properly validated by demonstrating the discriminatory ability and reproducibility. Formulations with intentional and meaningful variations with regard to particle size is a good testing sample to verify the IVRT method is discriminatory. For the IVRT result, because the product is for injection, The data should be provided as percent cumulative in vitro drug release, and the equivalence can be demonstrated using model-independent similarity F2 factor. To conclude, injectable drug substance suspensions are formulated without release modifiers, and the drug substance is the only insoluble component in the product. An in vitro B approach is based on the understanding of the critical quality attributes of the product that affect bioequivalence and relies on totality of evidence. The particle size distribution study and the in vitro drug release study should contain sufficient information regarding method development, method validation, and study result. Challenge question number 1. Is this statement correct? The particle size distribution method should be validated with regard to precision and robustness. Yes, this statement is correct. Challenge question number 2. Which of the following statements for the IVRT study in the PSG for injectable drug substance suspension are true? A. IVIVC is required to justify the used IVRT study. B. No method development and validation report need if the dissolution study information can be found in the FDA's dissolution methods database. C. The dissolution study information in the FDA's dissolution methods database serves as a starting point, and applicants need to submit sufficient method development and validation reports. D. The used IVRT method should be able to discriminate the critical quality attributes, for example, particle size. The correct answer is C and D. The dissolution information in the FDA's dissolution method database serves as a starting point, and applicants still need to submit sufficient method development and validation reports. The used IVRT method should be able to discriminate the critical quality attributes, for example, particle size. I would like to give thanks and acknowledgement to Dr. Bing Qing, Dr. Yan Wang, Dr. Darby Kozak, and Dr. Robert Leinberger. I also want to thank our internal collaborator, Dr. Xiaoming Xu and Dr. William Smith, as well as our external collaborator, Dr. Diane Burgess. Thank you for your attention. Please don't hesitate to reach out for any of the questions. Also, I will be back shortly to answer any of your questions in the panel discussion.
Thanks for the kind introduction. It is my pleasure today to discuss with you our recommendations in the product-specific guidance for peptide products, especially for the risk-based comparative immunogenicity assessment and impurity profile assessment. Our learning objective for today is number 1, after the talk, you should be able to understand and describe some of the immunogenicity risk and considerations for peptide products. Then understand and summarize some of the comparative analysis that may be included in a PSG for peptide products. And finally, you should be able to correlate the types of product-specific guidance recommendations with the different immunogenicity risk of a peptide product. Next, I would like to start off by providing a brief overview of product-specific guidances, or PSGs. So the agency provides the current recommendations for demonstrating therapeutic equivalence through these PSGs. This is important for developing generic drugs because generic drugs are approved based on the findings of therapeutic equivalence to the reference listed drugs, or also known as RLDs. Because of the potential immunogenicity risk of the peptide therapeutics, the risk of each peptide can also differ. We will recommend appropriate studies in the PSGs based on our current understanding of a product's potential risks. Of course, when our scientific understanding changes, we will use the PSG to communicate and update the appropriate recommended studies. And later in this talk, I'll give you an example of how we've done this with the vasopressin PSG. So peptides, they are typically shorter than biologics or proteins, and they typically have less immunogenicity risk. However, it is still just a shorter form of a protein if you think about it, and the same scientific concern and reasoning can also apply to peptides. And when we're looking at the immunogenicity risk, we're looking at the following considerations. First, from the peptide product considerations, which may include these following aspects. One of them is related to peptide size. So the longer peptides may have higher immunogenicity risk compared to the shorter one because it may contain more HLA binding epitopes. The route administration should also be considered because certain routes, such as sub-Q injections, will be more risky than, say, something that's oral route. Now, if the peptide sequence is highly homologous to endogenous human protein, then the concern is that if you raise an antibody against the peptide, it may cross-react with the endogenous protein and cause deficiency syndromes. And finally, if the peptide has a longer than usual half-life, you could have a concern where you may have greater chance of generating anti-drug antibodies, hence increasing its risk. We also should look at the other considerations when we're assessing immunogenicity risk, such as patient population and the drug indication, whether it's indicated for patient with risky immunogenicity or immunity issues. Other clinical experience of the RLD can also be used, such as the level of ADA generated in the clinical studies or any severe adverse events that are found during the clinical study. So with all of these considerations combined, we can get some relative information about the potential immunogenicity risk of a peptide product. So in order to minimize the risk and ensure that the follow-on peptide products are not higher clinical risk, including the risk of immunogenicity, then the reference we will want to see the comparative analysis showing no clinical meaningful differences. And these comparative analysis would include both analytical testing as well as functional and cell-based assays. API similarance should be demonstrated for all generic products, not just for peptides. But for shorter and simple peptides, this comparison can be as simple as just comparing the primary sequence. But for longer structured peptides, additional analytical studies to compare the HOS or high order structures, such as secondary structures and aggregation states, should also be recommended. Impurity difference may impact the immunogenicity potential of a proposed generic. So for peptides with elevated immunogenicity risk, we'll recommend not only to conduct the impurity profile, which is typically done using LCMS, but if a peptide has a higher immunogenicity risk, such as the 5 peptides outlined in the synthetic peptide guidance, the adaptive immunogenicity assay will be recommended to assess the presence of MHC binding potential. And for certain peptides with high risk, the innate immune response assays, which could also be recommended on the whole peptide product. And please note that this comparison may be recommended independent of the findings from impurity profile differences, meaning that even if you don't see a difference in your peptide-related impurities, the innate immune response assay should also be conducted. So here's a slide that summarizes the different types of recommended studies in these peptide PSGs. And as you can see, not all peptides are treated the same. We will recommend studies based on the perceived risk of these peptide products. So if we start from the top of the list, we have few simple peptides such as octreotide simple injectable and brahmalanotide. Both are small peptides and they have a minimal risk. So we just ask for simple API sameness comparison as well as high order structure and oligomer aggregate study. Then we move down the list with peptides such as vasopressin and secretin have elevated risk. They are slightly higher, so we ask for some impurity profile comparison, but in general, we are not recommending the adaptive or innate immune response assay here yet. Of course, you could have also biological activity assays recommended for PSG such as secretin. And then there's the dasiglucagon, which is slightly higher above these other peptides when it comes to risk. So for those, we will ask for innate immune response on top of the recommended studies. And then if you have something that's semaglutide, pretty much on par with the other 5, when it comes to risk, on par with the other 5 peptides that are outlined in the synthetic peptide guidance, we'll recommend all of these studies that are described in the synthetic peptide guidance. So next, I'll take a deeper dive into the PSG for these 3 peptide products, vasopressin, dasiglucagon, and semaglutide. Vasopressin is a non-amino acid polypeptide hormone that causes contraction of vascular and other smooth muscles. So initially, we had concern with this product having the possibility of developing anti-vasopressin antibodies after chronic dosing. So we recommended during the review of these applications for new impurities or immunogenicity assessment of new impurities that are present in the proposed drug product, as well as impurities present at an elevated level compared to the ROD. So essentially, we're treating these at a higher risk consideration and ask for the immunogenicity assays. However, after the approval of the 505(b)(2) vasopressin, we noticed that there is a difference in how these are internally. There's difference in how we review the vasopressin and as well as the B2s. We internally initiate reconsideration of the review practice for the vasopressin products. And after the discussion and internal review, we find out after further studying of the post-marketing experience of the marketed vasopressin product, we arrive at the conclusion that this product is considered to have a relatively low immunogenicity risk. So in our PSG, we also reflected this, that in general, immunogenicity risk assessment for impurities are not recommended or not necessary. So in February 2022, we developed and published the vasopressin PSG to communicate this change in initial assessment and further clarify the comparative studies needed. So instead of recommending conducting adaptive and innate immunogenicity assays, the PSG will only recommend conducting API sameness, impurity profile, aggregation profile comparison. Now, unless there's anything new, unusual new impurity or aggregation state, or highly elevated impurity or aggregation state in the proposed generic product, the immunogenicity assessment will not be requested. And next, dasiglucagon. Dasiglucagon is a 29 amino acid human glucagon analog peptide. Dasiglucagon is similar to glucagon sequence and indication. The reference is a synthetic product that is indicated for emergency use. So immunogenicity risk of the peptide appear to be lower because it is based on the ROD's labeling, where less than 1% of the patients developed anti-drug antibodies in the clinical trials. Because the risk of the diacylglucagon is slightly lower than glucagon and the other peptides outlined in the peptide guidance, the synthetic peptide guidance, the PSG reflects this by recommending comparative impurity profile studies without mentioning the absolute need for adaptive immunogenicity study unless upon the agency assessment an impurity of concern is discovered. However, other studies, such as the innate immune response assays on the whole product is also still recommended, along with all the other comparative studies, such as API sameness, including the biological activities. Finally, semaglutide. Semaglutide is a 31 amino acid GLP-1 agonist peptide. It is similar to liraglutide in sequence, also for the intended indication for the same patient population. So like liraglutide, it is a homologue of a non-redundant endogenous hormone and is chronically used through sub-Q injection. So because of these similarities to liraglutide, its immunogenicity risk we can say it's pretty much the same as the other 5 peptides, along with the liraglutide outlined in the synthetic peptide guidance. So therefore, in the PSG of semaglutide, we recommend the sponsor to follow the recommendations outlined in the synthetic peptide guidance. So although the peptide guidance is specific to the five peptide products listed in the guidance, scientific principles and elements of the synthetic peptide guidance may be applied to other higher risk products such as semaglutide. And for those unfamiliar with this guidance, it basically provides the recommendations on how to address the potential immunogenicity risk for peptide-related impurities. It's specified for 5 high-risk products, including liraglutide, tetraglutide, nasiratide, and glucagon. So in summary, different risks of peptide products will have different sets of studies recommended through the PSGs. The higher the perceived risk or immunogenicity risk of a peptide, the more recommended comparative and immunogenicity studies will be outlined in the PSG for that peptide. PSG reflects the current agency's scientific thinking on the most appropriate methodology for developing drugs and generating evidence supporting therapeutic equivalence. However, it is not static. The agencies can update the PSG when current thinking evolves or additional scientific knowledge gained. And lastly, please reach out to us through control correspondence or pre-ENDA process to discuss any proposed scientific studies that may deviate from the PSG recommendations. I'd like to end the presentation by acknowledging the following people who have helped me develop and organize this presentation. So now let's move on to the challenge questions. Question 1, true or false, PSG only contains recommended studies for demonstrating bioequivalent studies for generic products. The answer is false. PSG not only contains bioequivalent studies, but other studies as well to not demonstrating just bioequivalence, but also therapeutic equivalence of a generic product. Challenge question number 2. Which of the following statements is not true? A. PSG contains recommended studies to demonstrate product sameness for both pharmaceutical equivalence and bioequivalence. B. Immunogenicity risk assessment using non-clinical assays is recommended for all peptide products regardless of their risk. C. Adaptive and innate immune response assays are typically recommended for certain peptide products with immunogenicity concern. And D. Peptide products not covered by the synthetic peptide guidance may still reference parts of the recommendations outlined in that guidance. And the answer is B. Immunogenicity risk is not using non-clinical assays not only recommended for it's not recommended for all peptide products. It is depending on the risk of the peptide product. Thank you for your attention. If you have any questions, I'll try to answer them at the panel session.
Thank you for the great presentations. If you haven't had a chance to enter your questions in the Q&A chat pod, we remind you to do so at this time. We do have a few additional panelists we'd like to welcome to the panel. First, we'd like to welcome Dr. Daping Kui, Lead Pharmacologist, Division of Bioequivalence 1, Office of Bioequivalence and OGD, and Dr. Cameron Smith, Branch Chief, Division of Liquid-Based Products 1, Office of Lifecycle Drug Products, and OPQ. We do have some questions. The first few questions will be addressed to Dr. Xinan Zhang, and here is the first question for Dr. Zhang. For injectable suspension products, during in vitro drug release testing, what is the recommended temperature at which the dissolution media should be maintained?
Thank you for the question. Normally, we do not recommend a specific temperature for the dissolution media to be maintained. We recommend you provide the method development and validation data support your selected temperature. Thank you.
Thank you for responding to that question. We do have a few more questions for Dr. Zhang, and here is the next question. Regarding drug substance injectable suspension, to prove the discriminatory of the dissolution method, generic players should perform intensive experiments like varying active and excipient concentration, PSD, non-sync condition. Please suggest leftover parameters to study the discriminatory.
Thank you for the question. I think you provide a great example regarding the discriminatory study for the selected dissolution method. The study of discriminatory ability can be achieved by intentionally manufacture a few batches of the product with meaningful variations in formulation and manufacturing parameters. For example, particle size and drug loading. Um, we recommend you identify the critical product attributes that affect drug release and demonstrate that your dissolution method can be used to discriminate this potential difference. Thank you.
Thank you for responding to that question. Our next question for Dr. Zhang is the following. You mentioned about flocculation of particles. Does flocculation need to be considered for particle size distribution for a particle size distribution study? And also, if R&D data shows that flocks affect IVRT, will IVBE be done using flock size?
Thank you for the question. This is a great question. First, I would like to address the first part of the question. Does flocculation need to be considered for particle size distribution study? I want to answer this first question from two aspects: what to be submitted for particle size distribution study, and what data should be subjected to PBE analysis. In brief, when characterizing particle size of suspension formulation with flocculation feature, the general expectation is that applicants should characterize particle size under different conditions, capturing both flocculated and deflocculated particle. With regard to PBE analysis for establishing equivalence in particle size, only the condition that mainly capture the size of deflocculated particle, the primary particle, is needed. It is noted that the flocculation is a formulation characteristic, and it is mostly governed by formulation composition. Therefore, when a proposed formulation deemed Q1, Q2 to the reference formulation, the main source for potential difference in flocculation behavior is the particle size of the primary particle. As such, applicants should measure the PSD under continuous sonication, which allow better capturing the contribution from primary particle population for PBE analysis. To conclude, the flocculation behavior should be captured during particle size characterization, but only the primary particle is subjected to PBE analysis. For the second part of this question, I want to highlight the purpose of IVRT and particle size characterization are different. IVRT is a general performance testing. It should be noted that it is hard to have a condition that all particles are flocculated or deflocculated. So for the IVRT study, applicants can develop and validate conditions per reproducibility and discriminatory ability, and the condition will likely have both flocculated and deflocculated particle present. Thank you.
Thank you for responding to that question. We have one more question for Dr. Zhang, and here is the question. Is a separate method development report for particle size required if a method validation report is present and method development information is included in the product development report?
The short answer is no. So when we talk about the method validation and the method development, it can be one report in general. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that came in from Dr. Eric Pang. And here's the first question. The PSG did not recommend an impurity profile, adaptive, and innate immunogenicity studies for octreotide injectable. Under what circumstances does the agency request these immunogenicity studies for octreotide injectable?
Thank you for the question. So for octreotide injectable, simple injectable, is our understanding that it is a solution, it's a product with low immunogenicity risk. So typically these will not be required to have immunogenicity studies to be submitted upon review, unless there's some impurities. As always, if there's something in there that shows to raise the impurity immunogenicity risk, for example, or something that's highly unusual impurity, or if the formulation could, well, it depends. I think most of the time it's just that if there's a very, very different impurity or something that's very highly elevated impurity, those will be something to be a concern about. But still, it depends on what kind of impurity it is.
Thank you for responding to that question. We have another question for Dr. Pang. And here's the question. Regarding innate immunogenicity, is it adequate if the test drug product demonstrates lower innate immunogenicity potential compared to the reference listed drug, or should it demonstrate equivalence with the RLD?
Thank you for the question. So let me just clarify. So the purpose of demonstrating immunogenicity risk is not to demonstrate equivalence of your testing or data, but more of there is not a higher than the reference listed drug when it comes to risk. So it's preferable that your product demonstrate no higher risk than the reference listed drug. Thank you for the question.
Thank you for responding to that group of questions. Moving on to the next panelist, we do have a question that came in for Dr. Daping Kui. And here is the question for Dr. Kui. What is the recommended duration dissolution runtime for in vitro drug release testing for injectable suspension products?
Thank you for the question. Sorry, I was on mute. Yeah. So the short answer is that no fixed duration for the recommended IVRT testing for all the injection suspension product. It's case by case, drug product specific, and I think the general rule is that when you conduct the IVRT, you should ensure that the duration you use for your study is sufficient to have a complete release for your drug products in the testing in your IVRT study, and also you should meet the requirement for make your testing to be discriminative. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Dr. Cameron Smith. Here's the first question for Dr. Smith. FDA has asked applicants to perform innate immunogenicity assessments using a sample of drug product batches at or near release and at expiry. What is the FDA's rationale for asking for this to be done? Wouldn't the at expiry provide the worst case for testing?
Hi, thanks for the question. Yes, I think I agree that the at expiry testing would likely be the worst case scenario. But the reason we ask for on our near release as well is because if there is an innate immune response modulating impurity that is stability indicating, that would help us get at that and elucidate whether or not that is the case. So, for example, if you had a leachable that was leaching from the container closure system, that would likely be a stability indicating impurity. And so you may, if that impurity induced an immune response, you may see a difference in the innate immune response between the release samples and the samples aged to the end of the shelf life.
Thank you for responding to that question. The next question for Dr. Smith is the following. Do you recommend using LC or LCMS data when comparing impurities in vasopressin and other synthetic peptides?
Yeah, so generally speaking, as the complexity of the peptide structure increases, we are more likely to recommend LCMS data or some sort of two-dimensional mass spectrometry and chromatography type data. It helps us get at two things, whether or not the analytical method is adequately separating all of the peaks that are present, all of the impurities that are present, because you can use the second dimension of mass spectrometry to elucidate peak purity. And so that's why we recommend that in most cases, so that you can adequately characterize the peptide profile that's present in your product.
Thank you for responding to that question. We do have one more question for Dr. Smith. Here's the question. Is there a possibility that for adaptive immunogenicity, the agency would require the testing of the whole product and not only individual peptide-related impurities?
No, generally speaking, the adaptive immune response testing is only conducted on purified impurities. We likely would not ask for that to be conducted on the formulated drug product. And so this is in contrast to what we'd expect for the innate immune response assays, which are conducted on the fully formulated product.
Thank you for responding to that question. We've got time just to try to get one more question in. We have a couple of questions left for Dr. Xingnan Zhang. And here's the question for Dr. Zhang. Is sync condition required for IVRT studies?
We acknowledge the drug substance solubility is generally very low in drug substance injectable suspension products, and we understand sometimes a non-sync condition is used for improvement of method discriminatory ability. Therefore, sync or non-sync condition either way may be acceptable as long as you provide sufficient justification. Thank you.
Thank you for responding to that question. And that's all the time we have for questions in this panel. A huge thank you to our speakers and panelists for answering numerous questions that came in from our audience. We'll move right into our final session, which is session 4 on noteworthy complex generic drug approvals, multi-phase systems. Our first presenter is Dr. Quickly Fan, Pharmaceutical Science, Division of Liquid-Based Products 2, Office of Life Cycle Drug Products in the OPQ. Dr. Yuriko Haragaya, Senior Staff Fellow, Division of Bioequivalence 2, Office of Bioequivalence and OGD. Then we welcome Dr. Ben Chen, Senior Chemist, Division of Therapeutic Performance 1, Office for Research and Standards and OGD. And our final presenter is Dr. William Smith, Research Scientist, Division of Product Quality Research, also Office of Testing and Research and OPQ. Now, please join me to welcome our first presenter, Dr. Fan.
Thanks for the introduction. Hello, everyone. Thank you for joining us today. I will focus on what's new in the CMC part of these two ophthalmic emulsion products. Dr. Haragaya will focus on the BE part. Learning objectives. Highlights of new draft PSG on cyclosporine and the new nuclear magnetic resonance NMR method for quantifying the drug distribution of difluprednate in different phases. The background of my talk. The PSG provides the in vitro BE option, which following criteria need to be met. First, Q1, Q2 sameness, which is the test and RD formulations to be qualitatively and quantitatively the same. Second, acceptable comparative physical-chemical characterization, Q3, including drug distribution in different phases. Third, acceptable comparative globule size distribution, which is now in vitro B study 1 and B study 2 RVRT. Again, I will focus on the first two criteria. First, what's new in current draft PSG for cyclosporine ophthalmic emulsion? This current draft PSG was published in August 2022, exactly 1 year ago. The major new part of this PSG is the footnote 3 on Q1, Q2 sameness. It's so important. Let me just read it out loud. For ophthalmic drug products, FDA has determined that as a scientific matter, any qualitative or quantitative deviations from the IOD, even in inactive ingredients listed in 21 CFR, should be accompanied by an appropriate in vivo BE study or studies. Hopefully you have already noticed that and carried it out in your development work. For the part of parameters to measure, now the comparative globule size distribution moved to the in vitro BE study 1, again will be focus of Dr. Haragaya's talk. If you are interested in the background information of this PSG and other details of this PSG as well, I highly recommend this journal paper titled as Product Quality Research for Developing and Assessing Regulatory Submissions for Generic Cyclosporine Ophthalmic Emulsions. This is published in the AAPS journal in January 2023 this year. Also, there is device information in this PSG as well. Second, the NMR method for quantifying the drug distribution of difluprednate in different phases. This research work has been done by our colleagues in OTR of FDA, and the work was published in molecular pharmaceutics last year. Actually, it was the cover feature of the July issue of that journal. From the cover photo here, you can see the vivid cartoon depicting API distributed in oil and surfactant phases, plus their well-separated peaks in the spectrum. Very briefly about the method. The method uses certain dilutions so that API in the surfactant phase is more resolved. And here you can see these well-separated distinct peaks of APIs in different phases. Black peak represents API in the water phase. There are two red peaks. The left one represents API in the surfactant phase, and the other red peak is API in the oil phase. Here is the brief description of the spectral and method. So what are the benefits of this new NMR method for drug distribution? First, it's non-invasive. Second, as I just showed you, distinct peak of API in each phase in the spectral is achieved. Third, it's easy to measure API distribution in different phases by integration. How about the mass balance? Here you are. In water phase, it's about 1.8%, very similar to the API's solubility in water. In surfactant phase, there is 35%. Of course, the majority API is in the oil phase, 59%. In total, it's about nearly 96%. It's very good mass balance results. If you want to use this method, it needs to be validated, and we recommend it as a one-time study. What about the other method for the drug distribution? Here you can see this one is the ultracentrifugation, and this is the ultrafiltration. Also, the third one will be dialysis. Here's my challenge question. Very easy. Which method for drug distribution study in different phases is non-invasive? Is it A, ultracentrifugation, or B, ultrafiltration, or C, NMR, or D, dialysis? Well, it's very straightforward. It's C, NMR. The summary of my talk. First, I presented highlights of new draft PSG for cyclosporine ophthalmic emulsions. And then I briefly described the NMR method for quantifying the drug distribution of difluprednate in different phases. This is a non-invasive method. Finally, I want to thank my branch chief, Dr. Helen Zhang, Division Director, Dr. Pahala Simamora, Associate Director of OLDP, Dr. Andre Rao, for reviewing my slides, and also Dr. Kang Chen, who is the PI of this method development work, answering a lot of my questions. Last but not least, SBIA organizers, thank you very much. With that, thank you for your attention, and I'm looking forward to your questions.
Thank you, Dr. Chu Xinfen, for very informative CMC portion of presentation. Hello, everyone. Thank you for joining us today. My name is Yoriko Harigaya from Division of Bioequivalence 2. In my presentation, we are going to take a look at two ophthalmic emulsions from the bioequivalence perspective. This presentation will tell you the differences between two cyclosporine ophthalmic emulsions in globule size distribution studies, how to select the membrane in in vitro release test for cyclosporine and difluprednate ophthalmic emulsions, how long in vitro release tests be performed, what is discriminative in vitro release test. As Dr. Fan mentioned, current draft product-specific guidance provides an option of in vitro studies for cyclosporine and difluprednate ophthalmic emulsions. My talk will focus on two in vitro studies, globule size distribution and in vitro release test. First, I will share the recommendations on cyclosporine ophthalmic emulsion for globule size distribution studies. Why globule size distribution study recommended in PSGs are different in two cyclosporine ophthalmic emulsions? For cyclosporine ophthalmic emulsion 0.1%, PSG recommends evaluation of D50 and span to demonstrate bioequivalence. However, cyclosporine ophthalmic emulsions 0.05%, PSG states D50 and span may not be sufficient and recommends evaluation of Earth mover's distance or other suitable metrics. It is because cyclosporine ophthalmic emulsion 0.1% displays monomodal distributions. D50 and span are reasonable parameters to be evaluated. While cyclosporine ophthalmic emulsion 0.05% displays multimodal distributions, thus D50 and span cannot describe the shape of distributions. Next, I would like to share the general recommendations for in vitro release tests for cyclosporine and difluprednate ophthalmic emulsions. These recommendations may not apply to your novel IVRT methods. How to select membranes for in vitro release tests? Since selection of the membrane will significantly affect drug release kinetics, it is important to test different types of membranes. Also, evaluating multiple replicates of each membrane will help identify the reproducibility. Pore size of the membrane should separate drug from oil globules. Thus, ideally, pore size is smaller than the globule size and larger than size of micelles. We also recommend to evaluate the inertness of membrane to minimize the membrane adsorption. Mass balance study identifies whether your in vitro release test measures drug release from oil globules. Most oil globules ideally stay in the donor compartment when drug and micelles are released to the receptor compartment. How long in vitro release tests should be performed? For ophthalmic emulsions, you may see a biphasic release profiles, such as aqueous and micellar phase will be released rapidly. Then slow drug release from the oil phase may be observed. In vitro release tests should capture the entire drug release profiles. So how long it should be performed? Ideally, in vitro release tests be continued till you observe a complete release, which is at least 85% of drug label claims. Or continue the study till no significant increase observed over 3 consecutive time points. If your in vitro release test does not reach 85%, we recommend that you submit justifications for not reaching complete release. What is discriminative in vitro release test? In vitro release test should capture the gradual drug release from the oil phase. Thus, study duration can be long. From the bioequivalence perspective, in vitro release test does not need to mimic the physiological condition. Therefore, no need to predict the bioavailability. Your in vitro release test needs to discriminate different quality attributes or manufacturing process, such as different globule sizes or other physicochemical properties. Also, we generally recommend to use F2 similarity factor for comparison of 2 drug release profiles. F2 will compare the entire release profiles, not just one time point. And ideally, variability be reasonably low to have an adequate sensitivity. For instance, in this figure, if the red profile with the long error bars is a control from multiple batches, formulations altered 1 and altered 2 were considered not discriminative. This control may include test or reference batches evaluated in the method validation and pivotal studies to understand the batch-to-batch variability. In the bottom figure, in vitro release test displays low variability, and you can see the separation between control batches and altered formulations. Thus, this IVLT discriminatory ability is more desirable. Here is a poll question. Which one is not needed to be evaluated in the donor and receptor compartment in the mass balance study for ophthalmic emulsions? A. Active pharmaceutical ingredient. B. Oil. C. Diluent. D. Surfactant. The answer is C. Diluent. In summary, different cyclosporine ophthalmic emulsions may have different shapes of globule size distributions. For in vitro release tests for ophthalmic emulsions, your membrane should separate drug from oil globules. Mass balance study will help identify the reasonable in vitro release test. For discriminative in vitro release test, the entire drug release profile should be compared, not just a single time point. I would like to acknowledge countless support from Honlin Zhang, Xiaozhen Zhang, Xinlan Li, Heesun Chen, Den Zhang, Xiaomin Xu, and SBIA Organizing Committee. Thank you for your attention. This concludes my presentation. Now I would like to hand it over to Dr. Bin Qin for his presentation of Amphotericin B liposome.
Hello everyone, my name is Bin Qin. I'm a Senior Staff Fellow in Office of Research and Standards at the Office of Generic Drugs. My talk today is about Amphotericin B liposome and its product-specific guidance revision. In this presentation, I will first describe the approved Amphotericin B drug products and discuss potential challenges to develop generic Amphotericin B liposomes. Next, I will explain FDA's latest revision to the product-specific guidance on Amphotericin B liposomes. And lastly, I will illustrate GDUFA research on this product. Amphotericin B is a protein antibiotic first discovered in 1950s. It's an amphiphilic molecule. The monocyclic protein lactone ring of amphotericin B contains a hydrophilic polyo subunit and a hydrophobic protein subunit. Amphotericin B can bind to ergosteroids of the cell membrane of susceptible fungi. It forms transmembrane pores, leading to compromise of membrane barrier function. Ions can leak out, resulting in cell death. Amphotericin B can also bind to cholesterol components of the mammalian cells, with a binding avidity more than 10 times lower than that of ergosteroid. The binding to cholesterol is the main cause of toxicity. Amphotericin B is poorly soluble in water. It's presented as monomer or aggregates. Heat treatment can induce superaggregation of amphotericin B, reducing in vitro toxicity. As we know, amphotericin B has very low water solubility and membrane permeability. So many efforts has been made to develop drug products for parenteral route of administration. There were 4 approved drug products. Fungizone was the first amphotericin B drug product approved by FDA. It was approved back in 1966. Fungizone uses biosol detergent deoxycholates as excipients, and it can form micellar suspension when reconstituted in 5% dextrose solution. In 1990s, 3 lipid-based formulations were approved by FDA. They were Acetate, Amphotericin, and Emulsions. Acetate contains ribbon-like particles, and Amphotericin contains disc-like particles. Emulsions is 2 single-layer liposome formulations among the 4 approved drug products. The drug substance in Emulsions is intercalated within the membranes rather than loading into the aqueous core. This is very different from liposome products such as Doxil. Emulsions has been widely used for the treatment of fungal infections and other indications. The drug product has been included in WHO lists of essential of medicines, even though it may be difficult to assess the products in many countries. The global sales of Emulsions is about $500 million, and the US patent expired in 2016. There have been a lot of interest in generic production of this product. However, like other liposome injection drug products, development of generic version of amphotericin B liposome is not straightforward, and there are many potential challenges. For example, how to demonstrate bioequivalence and technical difficulties in manufacturing. Like other liposomal products, to demonstrate bioequivalence, conventional PK may not be sufficient. Additional supportive studies are needed. For manufacturing, a special consideration is the toxicity of the finished products. Manufacturing process can impact the product toxicity by change the aggregation status of amphotericin B in the lipid bilayer. So in this case, the toxicity assessment should also be considered when you establish bioequivalence of amphotericin B liposome. This slide shows you an example how manufacturing process can play a role in the in vitro toxicity of amphotericin B liposome. There were 2 follow-on amphotericin B liposomes products approved outside the United States. These 2 products were reported to have identical chemical compositions as Emulsions, but were manufactured differently. The result of comparative red blood cell assay shows that the drug concentration inducing half maximum potassium release, also known as TC50, for both follow-on products are substantially lower than that of Emulsions. This suggests that they have lower in vitro toxicity, or they have higher in vitro toxicity than Emulsions. Of note, one of the products, Amphotericin, has been withdrawn from market due to toxicity concerns. The agency has posted a guidance for industry to address topics related to liposome products, including CMC, human PK and bioavailability, and labeling documentation. Additionally, the agency has issued a product-specific guidance on Amphotericin B liposome to facilitate generic drug development. The PSG was initially posted in 2014 and revised for 2 times. The latest revision was made in 2020. I will talk about the latest revisions in the following slide. The current product-specific guidance on Amphotericin B liposome recommends in vivo studies and in vitro studies to establish bioequivalence. There are 2 changes in the 2020 revision. The first change was made on the in vivo study. Previously, a multi-dose steady-state PK study in patients was recommended in the PSG. This revision changed the study to a single-dose PK study in healthy subjects. This change was made based on new healthy subject information provided by generic drug industry through control correspondences and pre-ANDA meetings. Another change was adding in vitro red blood cell assay and measurement of state of associated Amphotericin B and the lipid bilayer in the in vitro studies. This change was made based on finding from GDUFA research project. In the next few slides, I would like to provide you an overview of GDUFA research for Amphotericin B liposome. This is a list of GDUFA research projects that have been conducted for Amphotericin B liposomes. I would like to share with you some of the key findings from the last 2 contracts, as this data help us to better understanding what are the critical process parameters of Amphotericin B liposome manufacturing, and how these parameters influence the in vitro toxicity, as well as what type of categorizations can be used to inform the in vitro toxicity. In the research projects, a 4-step process described in the US patent was used. These 4 steps include Step 1, mixing lipids and drug and organic solvents. Step 2, removal of organic solvents by spray drying. Step 3, hydration of the spray dry powder and microfluidization to form liposomes. And Step 4, lyophilization to a dry powder as the final product. The patent protocol has no comments on the extended heating step and only reference a 40 to 60 minutes incubation at 65 degrees Celsius during lipid hydration in Step 3. Through accidental observation and literature survey, the research team identified that an additional heat treatment, so-called curing, is needed to produce formulations that have similar in vitro toxicity profile to Emulsions. So the effect of the curing step was extensively investigated throughout the projects. The manufacturing process was evaluated using quality by design approach, 9 critical process parameters, and 5 critical quality attributes were used. Figure on the right shows the variable importance plot. This plot presents the impact of all process variables on all quality attributes simultaneously. The most important variable is the curing temperature, followed by quantitatively formulation difference, and lastly by liposome processing parameters. To study the potential impact of curing on different steps of the process, liposome samples that were generated with curing after Step 1, solvent mixing, Step 2, spray drying, and Step 3 microfluidization, respectively. UV-Vis methods is a commonly used method to study the aggregation status of free amphotericin B, and it's indicative of aggregation status change of amphotericin B. So this method was adapted to categorize the liposome formulations. The red dash on the right figure presents the main peak position of Emulsion 321 nanometer. As you can see here, only when the formulation was heated in step 3 did the main observation peak shift to 321 nanometer. This is consistent with that of Emulsion. Red blood cell assay was also conducted to evaluate the in vitro toxicity of these samples. Only the samples heated after microfluidization, step 3, show a significant reduced toxicity. This tells us the curing incubation should be introduced after step 3. Curing temperature and curing time can also have an impact to the product. Liposome formulations cured at different temperatures and time were also prepared. The spectrum on the left shows as curing temperature increase, main peak underwent a blue shift from 325 to 350 nanometers to 321, 322 nanometers. This suggests the aggregation status of Emulsion undergoes change from the loose aggregates such as monomer or dimer to a tight aggregate such as oligomers. The figure on the right shows the change of main peak position as a function of time under different temperatures. You will see here only a curing temperature above 50 degrees Celsius can achieve similar main peak position as the Emulsions. And here, the study also indicated that the timing of curing is also critical. The in vitro drug release of liposome formulations of those samples were also studied using a method developed by University of Michigan in a previous grant. The cumulative drug release profiles over first 80 minutes and over 25 hours are presented on the right. The release profile of Emulsion is in the blue color, and the uncured control sample is in the gray color. You can see here a general trend is when an incubation time is fixed to 6 hours, higher curing temperature samples have slower drug release rate, and the profile is closer to the release curve of Emulsion. To better understanding the drug release behavior, two first-order release kinetics models were used to fit the release curves. When the first one-pool release model was used, we can see there were good linear correlation coefficients for all the curves of initial 75 minutes data. And we also observed high curing temperature resulted in a slower release. However, this model poorly fit the full 24 hours release curve. We then use first-order two-pool release model to fit the curves. We can get a good fit for all the complete hour data set. This suggested the two-pool model can better categorize the release kinetics. This model indicated that amphotericin B may exist in two different aggregation forms, loose and tight. This is also deduced from UV-Vis analysis. Figure on the right shows the changes in time of the two aggregate pools. This is expressed as concentration. It shows that the tight aggregation pool, pool B, increases at the expense of loose aggregate pool, pool A, while the total drug concentration remains fixed. Potential correlation between the in vitro toxicity and in vitro release kinetic parameters were explored. The results below show that there were good linear correlation between normalized in vitro toxicity and the initial rate constant from the 1-pool model and the CA-CB ratio from the 2-pool model. There were also good correlations between normalized in vitro toxicity and a peak position and main peak ratio. In this slide, I would like to provide a quick summary about the GDUFA research I just presented. Curing was found to be very important for reducing the toxicity of amphotericin B liposome formulations. As the curing progressed, amphotericin B can change from the loose aggregate to a tight aggregate. You can find this shift from the UV-Vis methods and in vitro release testing. The two physicochemical and analytical methods, UV-Vis and IVRT, correlated very well to in vitro toxicity. As a side note of this presentation, the first two generic amphotericin B liposome products were approved in 2021 and 2022 respectively after the 2020 product specific guidance revision. Here are some take-home messages of my presentation. GDUFA research and information submitted by the generic drug industry has been very helpful to the guidance revision on amphotericin B liposomes. Manufacturing process play a very important roles in product toxicity. The product toxicity can be informed via physicochemical characterizations, in vitro drug release, and in vitro potassium release assay. We have two challenging questions. This is challenge question number 1. Is this statement correct? The in vivo bioequivalence study should not be conducted in healthy volunteers, as amphotericin B liposome is toxic. A. This statement is true. And B. This statement is false. Please take a few seconds to think about this question. The correct answer is false. Based on the new healthy subject information provided by industry, it's deemed safe to conduct bioequivalence studies in healthy volunteers. Challenging question number 2. Which of the following supportive comparative physicochemical categorization is not relevant to bioequivalence of amphotericin B liposome? A. Liposome size distribution. B. In vitro red blood cell potassium release assay. C. State of associated drug and lipid bilayer. And D. Internal environment. The correct answer is D. Internal environment. For amphotericin B liposome, the drug substance is intercalated in the lipid bilayer of the liposome. So the internal environment is not relevant to the bioequivalence of this drug product. With that, I would like to conclude my presentation. I would like to take this opportunity to thank my team leader, Yan Wang, my supervisor, Dabi Kozak, and my office director, Rob Weinberger. Thank you for their great help in the preparation of this presentation. Thank you for your attention. If you have any question, I will be happy to answer your question in the panel discussion session. Thank you.
Hey everybody, this is William C. Smith. I'm a research scientist over at the Office of Testing Research. Today I'm going to talk to you a little bit about some of the lab work we did on phytonadione injection relating to how the thermodynamics and self-assembly of the system work. So hopefully during this talk, you'll learn something about how to recognize the difference between macro, nano, and microemulsion properties. how to identify some of the key aspects of the manufacturing of these different formulations, and hopefully at the end you'll learn something about how to describe the difference between thermodynamically and kinetically stable systems. Okay, so let's look at our system of interest here, which is Phytonadione injectable formulation. So Phytonadione injectables have three principal components: Phytonadione, which is an oil-like immiscible API, vitamin K, a polyethoxylated fatty acid derivative, which is the surfactant, acts like the emulsifying agent or a soap or a detergent, and it's all dispersed in an aqueous phase. Now, the reference listed drug and the reference standard have subtly different ways that they denote the dosage form. The first being an aqueous colloidal solution, and the latter being an injectable emulsion. Now, you might think because we have an immiscible oil phase in an aqueous phase and some kind of surfactant that we could, choose either of these two options. But the fact that there is this subtle difference in the dosage form and the question of the dosage form really has some interesting implications on the regulatory pathway. So this subtle difference in the dosage form actually has an implication on the bioequivalence pathway, where something that's low risk, like a solution, if deemed to be qualitatively and quantitatively similar to the other product, is assumed to be therapeutically and bioequivalent. This is in contrast to something like an emulsion, where even if qualitatively and quantitatively similar, things like the manufacturing process can impart different physical and chemical properties to the formulation, which may necessitate additional in vitro testing or a different suite of in vitro testing in order to establish or demonstrate bioequivalence. So the question isn't really, is this a solution or is this an emulsion? The question is really, is this a thermodynamically stable system or a kinetically stable system? Here you can see we have emulsions up in the high particle size range, nano emulsions in the center here in the 500 to 100 nanometers. And then at the end, our 100 to 10 nanometer particle size is the micro emulsions. And so you can think about these different forms of the dispersion as kind of in a continuum, both in the components, so they can be made of all the three same components, maybe in different ratios or maybe at the same ratio. But then the processing equipment or the conditions that are used to manufacture the particles or the dispersions can actually change the particle size. And so the interesting thing is that the particle size is what is imparting all of these differences you see here in the appearance, so things like turbidity, whether or not it's opaque, whether it is an optically transparent or isotropic system, like you see here with the thermodynamically stable microemulsion. So I'm going to take just a slight sidetrack here to kind of make a point. And that is, some of the keen eye amongst you might have seen that I'm referring to the smallest particle size you saw there as a microemulsion. So according to the Encyclopedia of Colloid and Interface Science, a microemulsion is basically a micellar system which is transparent or translucent, so optically isotropic, and can be described as a swollen micelle. And so this is really just kind of a holdover of kind of when this was introduced in colloid science. So back in the 40s and 50s, when all of this kind of started, we had swollen micelle and microemulsion, and really the nano revolution started in the last 30 years. And so nobody was really using that terminology at that point in time. And you can see here the very first publications on microemulsions, where they started to go away from the swollen micelle system, which was kind of typical back into the 1800s. So if you remember back to before our detour, I was saying that the composition and the processing can impact the particle size, which can kind of inform what state our dispersion is. So if we look on the left side here, this is a ternary phase diagram where we have 100% of the composition of our three different phases, so surfactant, water, and oil, representing these vertices. And you read this basically as this vertice represents 100%, and then directly orthogonal to that line is 0% for that one phase. And so right in the center represents a 33%, 33% mixture of these things. And so how you can kind of think about microemulsion system is kind of existing in this where you just have, you know, your soap or your surfactant dissolved in water, and you are adding a little bit of oil into it. And so our microemulsion systems tend to live in this kind of edge region. Whereas our emulsion systems tend to be in kind of a lower surfactant regime with kind of a higher oil content. Now you can think about how to get your particles to smaller and smaller sizes in kind of two ways. First, you can make a bunch of micelles and then add some oil into them. And secondly, you can also think about taking the oil and breaking it up into smaller and smaller pieces and having the surfactant stabilize that surface. And so you can think that there's kind of this immense amount of energy at the oil-water interface. In order to reduce that energy, you can do something like add a surfactant or a surface actant agent, which actually decreases the interfacial tension and leads you to be able to create smaller and smaller particles. The other way that you can impart enough energy into the system to reduce the oil globule size is through just introducing a high energy process like mechanical shearing or increasing the temperature. So for your typical emulsion or nanoemulsion, you're taking your oil droplets and you're breaking them into smaller and smaller pieces using some kind of high energy process where you've reduced the size to where there's a sufficient amount of surfactant that can kind of stabilize that surface and kinetically trap it at that particle size. And so two main techniques that are used here are things like high speed homogenization or something like a microfluidizer, which uses high shear through high pressure changes. This is in kind of stark contrast to a thermodynamically stable system like a microemulsion where you only need a low energy input. So here on the right hand side we can see we're just basically dissolving a surfactant and oil stock solution into an aqueous phase, and it will slowly just disperse over time at ambient temperature and at low shear, so just with stirring. And it's important to remember that with the high energy techniques, you tend to only be able to get into that sub-micron range to around above 100 nanometers, whereas with the microemulsion system, you get into that kind of, 10, 20, 30 nanometer range. So another kind of critical aspect of the thermodynamically stable system is that it's really kind of process independent. So if you mix it slowly over the course of an hour, just dissolving it, versus you homogenize it over the course of about a minute and a half, you still end up producing the exact same dispersion. So in this investigation, we can take the path independence idea one step further. Where instead of adding our organic phase into our aqueous phase, like in the previous examples, we can add the aqueous phase directly to the organic phase and go through this kind of phase inversion process. So at an equivalent composition, what we end up seeing is that we kind of get this contradiction where we get two phase diagrams, one where we're pretty much always producing microemulsions, and the other where we're producing these opaque nanoemulsions. The important thing to note here is that when we're using an aqueous to organic addition, we do see a dependence in the particle size and the optical properties or turbidity based on the processing condition or the stirring. So the rate of stirring, the amount of shear, whether or not we use a homogenizer. Whereas when we use a stirring of any variety or a homogenizer in the organic to aqueous direction, we pretty much always get the same thing. So based on changing the formulation directionality and the processing conditions, we were able to form both nanoemulsions as well as microemulsions. Now the question becomes, at the composition of phytonadione injections, what is the most thermodynamically favored state? For any kind of a kinetically stable system like our nanoemulsion or like salad dressing, it's kind of trapped in this high energy state, and it wants to spontaneously minimize its energy, albeit maybe very slowly. And so for an emulsion system, if you are to disturb it either through giving it enough time and energy, it will tend to phase separate, say if you heat it or leave it for a long time in the refrigerator. This is in contrast to a microemulsion, where actually the microemulsion is the thermodynamically stable or favored state, where if you take two separate phases and leave them for a long enough time or impart enough energy, the microemulsion will spontaneously form. So the question is, if we give our systems enough time and or energy, are they going to split, demonstrating that they are not thermodynamically stable, or are they going to change from a nanoemulsion spontaneously into a microemulsion? So what does happen when we try to split our nanoemulsions that we formed by adding the aqueous into the organic? Well, if we incubate them for about 30 minutes at 70 degrees C, you can see here that we have two different particle size populations, around 15 nanometers here, and then something in the, say, 100 to 200 nanometer size range, which is imparting this opaqueness to our system. And then after that, we get one single particle size distribution in that 15 nanometer range that looks nearly identical to the microemulsions that we formed when we did the original directionality of adding the organic into the aqueous phase. So when we heat these nanoemulsions, they don't split, they end up turning into a microemulsion. So we talked about two of the kind of critical additions here. So adding our aqueous into our organic phase, adding our organic phase into our aqueous phase, and lastly, and maybe the most non-intuitively, we could just add our oil directly into a surfactant dissolved into the aqueous phase and then homogenize them. And so what this looks like is this, where we have an initial macro or nanoemulsion after homogenization. After 24 hours, we get this biphasic macroemulsion split, so it forms two phases. But given enough time, about 30 days at room temperature, our macro or nanoemulsion becomes biphasic and spontaneously forms a microemulsion, given enough time and energy. So what this tells us is that those nanoemulsions that we made are actually kind of a temporary intermediate where given an increase in the temperature or just enough time at the formulation composition of phytonadione injection, the thermodynamically favored state of the system is the microemulsion. Just a challenge question here too, to see if you're paying attention. True or false, microemulsions can only be produced using high shear mechanical processes. Is that A, true, B, false, or C, I have no clue? This, of course, is false. Okay, last question. Rank these dispersions in order of increasing particle size. Is it A, macro, micro, then nano emulsion? Is it B, nano, micro, macro? Is it C, micro, nano, then macro? Or D, none of the above? This happens to be C. Microemulsions are actually the smallest in particle size. And I just want to thank the whole Novel Complex Generics team who worked on this with me, and thank you all for listening.
Thank you all for the great presentations. We're going to be moving into our final Q&A panel of today, and we'd like to welcome some additional panelists. First, we would like to welcome Dr. John Zhang, chemist, Division of Liquid-Based Products 2, Office of Life Cycle Drug Products and OPQ. Dr. Hee-Sung Chung, lead pharmacologist in Division of Bioequivalence 1, Office of Bioequivalence and OGD. Dr. Kondakar Alam, senior pharmacologist, Division of Quantitative Methods and Modeling, Office of Research and Standards, and Dr. Salming Su, Division Director, Division of Product Quality Research, Office of Testing and Research and OPQ. Do you have some questions? Or 1 second. My question sheet is repopulating. Just 1 second. All right, there we go. And the first group of questions will be addressed to Dr. Quixie Fan. And here is the first question. According to the current draft PSG on cyclosporine, are deviations of pH adjuster allowed?
Thanks for the question. That's a good question. It is always allowed, but if the deviation within 5%, according to the footnotes, you don't have to submit the in vivo B study. If it's higher than 5%, well, you have to submit extra in vivo B study. Hopefully this answers the question. Thank you.
Thank you for responding to that question. We've got a few more questions for Dr. Fan, and here's the next question. For a one-time study on drug distribution by NMR, is method validation required?
Yes, it is required. That's a short answer, because drug distribution is such a critical property and directly related to the bioavailability. It's just a one-time study. However, to make sure the quality of your study, the results of your study, validation is required. Thank you.
Thank you for responding to that question. We've got a few more for Dr. Fan, and here's the next question, also on NMRs. What are the advantages of NMR methods comparing to other methods of drug distribution?
Again, thanks for the question. As I mentioned, the AMR method first and foremost is non-invasive compared to other methods. Second, it has these distinct peaks, if you still remember the spectra I presented, and easy to measure just by integrate. The mass balance also excellent. So those are the advantage of this NMR method compared to other methods. Thank you.
Thank you for responding to that question. Another NMR question for Dr. Fan. Can the NMR method also measure the globule size?
Thanks for the question. The short answer is yes. If you get a chance to read the paper of NMR, which I refer to, there's a certain section about how to use NMR method to measure globule size. If you have a further question, I would encourage you to contact the PI and corresponding author, Dr. Kang Chen. He will be glad to answer any question you have. At least he is very helpful to to me and to our group. Thank you.
Thank you for responding to that question. And one last question for Dr. Fan in this round. And here is the question. How can we make sure our formulation is good to go for ANDA submission?
Thank you for the question. That's a very good question. You can certainly use the controlled correspondence process to make sure your formulation is good to go. Just let FDA to check to assess your formulation before all your development work. That will certainly save a lot of your time and labor later on. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist. Got a few questions that came in for Dr. Yoriko Harigaya. And here is the first question for Dr. Harigaya. For any ophthalmic products that are non-Q2 to the RLD, can in vitro studies still be used to demonstrate BE?
Thank you for your question. The short answer is no. As current draft PSG for ophthalmic emulsion recommends, the test product and reference risk to drug has to be Q1, Q2 the same to choose in vitro option to demonstrate bioequivalence. And also, as Dr. Chu-Xing Fan mentioned, the current refused to receive standard guidance as well as cyclosporine ophthalmic emulsion guidance states exception excipient is not applicable to ophthalmic emulsion. Thank you.
Thank you for responding to that question. We do have a few more for Dr. Harigaya. And here is the next question. In the pivotal IVRT study for ophthalmic emulsions, do 3 batches need to be tested for both test and reference products?
Thank you for the question. The short answer is yes. We recommend you to evaluate 3 batches for each test and reference product to evaluate batch-to-batch variability. And also, this information help us understand the discriminatory ability of your IVRT method. Thank you.
Thank you for responding to that question. The next question for Dr. Harigaya is the following. For mass balance studies for ophthalmic emulsions, do both donor and receptor compartments need to be evaluated?
Thank you again for the question. So for mass balance study, we recommend you to evaluate both donor and receptor compartment to ensure that total concentration is close to 100% throughout the IVRT study duration. Thank you.
Thank you for responding to that question. The next question for Dr. Harigaya. Is it acceptable to use the Higuchi model to compare 2 drug release profiles for ophthalmic emulsions?
Thank you for your question. The short answer is no, because Higuchi model is for semi-solid skin topical product, and this model will evaluate only the early portion of drug release profile. However, ophthalmic emulsion, we would like to evaluate entire drug release profile. So we generally recommend F2 similarity factor. Thank you.
Thank you for responding to that question. Just a couple more questions for Dr. Harigaya. Here's the next question. In the IVRT discriminatory study, does altered formulation need to pass F2 value of less than 50 for ophthalmic emulsions?
Thank you for the question. That is a good question. So desirable, ideally, F2 value is less than 50 for discriminatory ability testing. However, it is not required. If your method is providing greater than 50 F2 value, but control batches has low batch-to-batch variability or inter-batch variability, and there is a clear differences between control and altered formulation, and it's reproducible, we may consider it is discriminative. Thank you.
Thank you for responding to that question. Just one final question for Dr. Haragaya. And here is the question. Is providing GSD data and serial dilution recommended for ophthalmic emulsions?
Thank you for the question. So it depends on the method you use. If you are using dynamic light scattering method, we recommend you to provide serial dilution to make sure your selected dilution provides stable globule sizes and polydispersity, and also make sure to have no interference from polymers. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions for Dr. Ben Chen, and here is the first question regarding potassium release assay. Is it adequate if the test drug product demonstrates a lower toxicity potential compared to the reference listed drug, or should it demonstrate equivalence with the RLD?
Thank you for the question. I think from a toxicity standpoint, a lower toxicity potential for the test product should be acceptable. However, additional consideration is that if this value deviate too much from the RLD, there may be additional concern whether other physical chemical property has changed. As we know, like the thermal treatment can also, if it's too long, can also make the impact the API stabilization and other property as well. Thank you.
Thank you for responding to that question. And the next question for Dr. Chen is the following. How does the heat treatment affect the toxicity of amphotericin B liposome?
Thank you for the question. Yeah, for the heat treatment, what we find during GDUFA research and also in the literature is that the heat treatment actually can have an impact on the aggregation status of amphotericin B inside the liposome bilayer, lipid bilayer. If you have extensive heat treatment, you can shift the aggregation status from a more loosely packed status to a more tightly packed status, which can result in less toxicity. Thank you.
Thank you for responding to that question, Dr. Chen. We do have one more question for you in this round, and here is the question. What characterization studies can be used to measure the toxicity of amphotericin B liposome?
Yeah, thank you for this question. To measure the toxicity of amphotericin B liposome, the most common way is to use the red blood cells to measure the potassium release. And there are some other categorization methods you can also use to interact to show, for example, use the UV-Vis methods as well as IVIT methods. From the GDUFA research, we found those categorization can also have a can indicate the toxicity. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that just came in for Dr. William Smith. And here is the first question. Is globule size distribution considered a CQA for aqueous non-lipid injectable emulsions?
Hi, yeah. So in general, for emulsions, you can consider globule size distribution critical as the manufacturing processes tend to impact the globule size distribution. And you can think about that as being important because of the impact that the globule size distribution can have on the drug distribution as far as the aqueous proportion to the globule or the oil phase concentration of the drug, as well as anything dissolved in, say, a micelle. But it is really kind of depends. So the difference between like an ophthalmic and an injection, say for IV administration, would be the impact of the globule size distribution on the bioavailability based on that drug distribution. So in general, we kind of say globule size distribution for those types of products tends to be critical, but it really depends.
Thank you for responding to that question, Dr. Smith. We do have a couple more, and here's the next question. With the different modes of preparation for a phytonadione injection, did you see any differences in product stability?
Thanks for that question also. There are two different types of stability, right? There's physical stability and there is chemical stability. So when you produce an isocompositional product that is in one type of dispersion, so the nanoemulsion form and the microemulsion form, the chemical stability, so whether or not the phytonadione is degrading in the presence of UV light, is something that can be controlled for. The physical stability of a microemulsion is extremely high because it's the thermodynamically favored form of the dispersion. So things like heating and cooling or freeze thaw tend to not have a very large impact on the product. And for the nanoemulsion, what happens is it basically, because it's not the thermodynamically favored state of the system, when you do those kind of stability testings, they just become microemulsions. So in general, the stability is basically that the thermodynamically favored state of the system, depending on the composition, is what comes out. So if you made a different composition, you could get it to be destabilized because you have too much oil in there. But at the composition, the formulation composition of that product, when you undergo stability testing, it tends to just form into the microemulsion state. Thank you for that.
Thank you for responding to that question. We're moving on to the next panelist. We do have a few questions came in for Dr. Hee-Sun Chung. And here's the first question for Dr. Chung. Why is FDA not including the in vitro option as part of the phytonadione product-specific guidance? And are there any challenges in the in vitro option to achieve bioequivalence?
Thank you for that question. I'm going to provide more general response here. And it is that if you want to propose an alternative B approach than what is currently recommended in the PSG, you always have the option to approach the agency with your proposal. If you're at the pre-ANDA stage, then you can consider pre-ANDA product development meeting, for example. And if you're qualified, there's also control approach in which you can also approach the agency in that way. And so I leave it there. Thank you.
Thank you, Dr. Chung. We do have a few more questions here, and here's the next question for these dosage forms. What are some suggestions to ensure a complete ANDA package?
Thank you for that question. So this is an area, especially with these complex dosage forms, oftentimes we run into incomplete information within the submission, and we may end up asking for additional information, sometimes through information request, if it's a very straightforward information. If not, we may end up issuing a discipline review letter, and this may potentially increase the review cycle. So some suggestions in this regard are For example, make sure that your method validation report is complete. And in general, especially when you're dealing with the study to demonstrate bioequivalence, then you want to use the reference product to validate your method. Of course, there's a certain experiments, for example, selectivity, where you may need to use a modified form of the test product, and inherently then you cannot use a reference product for that validation. But in general, do use the reference product to demonstrate that method is validated for this drug product. Another is be sure to submit the method development reports, because oftentimes we don't have a set method to say this method works for this product or not. So it helps us to understand how you ended up with that specific method for the drug product that you're testing. Also, those two documents. Another thing that we run into is that sometimes we see from the difference or discrepancy between the formulation of the test product versus the manufacturing details from the chemistry related documents. So if we see some discrepancies in that regard, then we may need to reach out to you to clarify that. And we cannot make, for example, Q1, Q2 determination without this clarification. So those are the things that I would watch out for. Another is time to time, some of these drug products may be drug device combination. And if that's the case, then you should provide the comparative analysis results, and you can refer to the guidance, comparative analysis and related comparative use human factors studies for drug device combination products submitted in ANDA. So that's also very important, and you should consider with respect to the presentation of your product, etc. I do want to add to one of the earlier questions regarding the formulation of Q1, Q2 sameness. I think someone asked about the cyclosporine and the pH adjuster difference. In that regard, you may very well be aware that the agency posted a guidance last year related to pH adjuster for the ophthalmic, otic, and parenteral drug products. So if your product has pH adjuster difference compared to the reference listed drug, then you definitely want to look at the guidance closely and what other options are there for your drug product, even if it's not a simple solution, for example. Thank you.
Thank you for responding to that question. We do have another question that came in for Dr. Ben Chen. And here is the question for Dr. Chen. Can I have a meeting or a controlled correspondence if I have an ophthalmic product that is non-Q1, Q2, for example, a pH adjuster difference?
Thank you for the question. Yeah, actually, I'm glad you asked this question. Yes. So the quick answer is yes. The applicant, however, they should discuss with FDA through communication pathways such as control correspondences or pre-ANDA meetings. So in general, an applicant, they may be recommend to do an in vivo B study to address this, but we do recommend the applicant should first discuss with FDA. Also, we are aware there is a pH adjuster guidance, right? I mean, the main message we want to mention here is that we recommend the applicant discuss with FDA first, and then to see what type of study needed and what type of justification needed to submit in the ANDA. Thank you.
Thank you for responding to that question. We have a question that just came in for Dr. Kandakar Alam, and here's the question for Dr. Alam. Can the agency comment on modeling and simulation-based approach for generic drug approval?
Yeah, very important question. I think that this question is relevant for other session as well. So we have seen increased interest in the model-based alternative bioequivalence approach in recent years from prospective and applicants. In many cases, the prospective and applicants proposed model-based virtual bioequivalence in lieu of PSG recommended in vivo BA study. In fact, FDA has approved generic diclofenac sodium topical gel where the end applicant used virtual bioequivalence instead of PSG recommended clinical endpoint study. So model-based generic drug approval is not a fairy tale right now. It is a reality. And other than virtual bioequivalence, model-based approach can also be used to justify the potential difference between test and reference drug product, and to justify that the difference is not going to impact the in vivo performance. For example, when you have the mechanistic model, you can use the model to justify your case that the potential difference in Q3 data of complex systems such as difference in the globule size or viscosity or IVPT profile may not impact the in vivo performance of your test formulation. So agencies open and supportive of model-based alternative approach, but I also want to clarify that model-based alternative bioequivalence approach should not be taken as an opportunity to get around from the regulatory requirements. Model-based approach rather provides an opportunity to overcome the challenges that we have seen or the applicant have seen with the in vivo BA study. So model-based approach still needs to satisfy the requirement of the applicable statutes and regulation that FDA has. We have from the agency, we have presented in different public workshop to share our regulatory thinking on the requirement of model development and validation. So I would suggest the generic applicants to use these resources if they are considering model-based approach. I would also suggest that prospective applicant can interact with the agency early through proper communication channel like pre and meeting request or CC to get feedback on their model-based approach before they submit the end applications. So hopefully my response is helpful to the audience to some extent. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we do have a question that came in for Dr. Xiaoming Su. And here is the question for Dr. Su. What are the alternate approaches to determine the drug distribution of the active drug in the nano emulsion?
Yeah, thank you for the question. It's a good one, and there's always alternative approach. So as Dr. Fan mentioned in his talk, that NMR is one methodology that can be used to measure the drug distribution in emulsions. But of course, there are other approaches that can also be used, for example, dialysis and centrifugation, or even ultrafiltration. So as long as the globule portion of the product can be separate from the non-globule portion of the product, and that you can actually analyze the content in each portion. And then by definition, you are measuring the distribution of the drug in different portions. So, but of course, we have to recognize that the methodology, each methodology has its own limitations. Not every technique is necessarily suitable for or has its own limitations. We need to recognize as part of the development and validation, need to be really careful in considering how to demonstrate that method it's suitable to demonstrate the intended purpose. Focus on the reproducibility, focus on the precision. It's going to be very critical. But other than what's mentioned in the talk as an MR, certainly the other few can also be utilized to support the demonstration of the bioequivalence. Thank you.
Thank you, Dr. Zhu. We're going to try to squeeze in one more last question that came in just right now for you. In a few of the presentations, it was mentioned some work done by FDA or sponsored by the FDA. Can we use a different method to support our application?
Thank you for the question again. Different method can be used. I think one important perhaps takeaway, if you've seen a lot of FDA's work or the contract or grants sponsored by the FDA, is we're really trying to providing alternative pathways or conducting research to fill the gaps. And some of the published work really is intended to share what we have accumulated understanding to the public, to the industry, so that you may adopt some of the method that has been developed. But by no means those are the method that has to be used or the only method that can be used. So certainly if there are alternative method that can also serve its purpose, certainly we are welcome to submit. I think as Hisam mentioned earlier, that there are different pathways that you can communicate with the agencies through pre-ENDA, through controlled correspondence pathways. So all of that can be very helpful. So, but of course, our research work, whether it's done at FDA or sponsored by the FDA through grant, it's through GDUFA research. So it's paid by the industry. So it would be beneficial for you to consider looking into that and to see how maybe beneficial to adopt in your development program. But thank you very much.
Well, that's all the time we have for questions in this final panel of the day. A huge thank you to our presenters and panelists for answering numerous questions that came in from our audience. We'd like to welcome Dr. Lei Zhang, Deputy Director of Office of Research and Standards and OGD, to provide some closing remarks from day one. Welcome, Dr. Zhang.
Thank you, Ray. Good afternoon, good evening, and good morning to all the participants across the world. I hope that you, like me, enjoyed every session of today's workshop. Time just goes by so fast. This is the 6th year that we hold this workshop. We clearly see a continued interest from our stakeholders in this workshop. We have more than 3,000 people registered from 86 countries. Today, we covered several types of complex products, including topical and transdermal products, nasal suspension and inhalation products, complex injectables, products with complex APIs, and more. We learned how research supports PSG and general guidance development, and eventually ANDA approvals. The scientific research founded by Generic Drug User Fee Amendments, or GDUFA, program builds an essential bridge from the scientific knowledge of the modern era. Science and research are critical to modernizing the generic drug program and the generic drug industry to continue to supply safe and effective generic drugs to the American public. As reflected by the title of this workshop, we are elucidating how we translate science to approvals, better translate some of the complicated science behind generic drug development into more generic drug approvals. Now we are close to the end of the first year of the GDUFA 3. We already know there are a number of enhancements under GDUFA 3 that have been implemented to further enhance efficiency, transparency, and gain more first cycle approvals. These include PSG enhancement on new goals for newly approved complex products, options to request PSG-related teleconferences and meetings, and additional scientific meetings with industry both prior to and post-submission, so that applicants can communicate with the FDA in a timely manner throughout the product development cycle for input on the development program to improve efficiency and enable better ANDA submission or resubmission. Enhancements also include the suitability petition process, including the establishment of new suitability petition goals, which will begin next month in fiscal year 2024. Tomorrow, We will have another array of exciting topics, including additional complex products, such as orally inhaled products and locally acting GI drug products. In addition, the last two sessions of tomorrow will cover enhanced processes, GDUFA research and assessment tools to support generic drug development, and also global collaborations to support efficient generic development and regulatory assessment. A new pilot industry meeting program will be presented. You do not want to miss it. Before I end, I would like to thank the session leads, speakers, panelists, and the subject matter experts who worked diligently together to design and deliver the content for this workshop, both today and tomorrow. I also would like to thank FDA Commissioner Dr. Califf for delivering the keynote speech this morning, and always our SBIA colleagues, Brenda, Ray, and others in moderating the sessions and coordinating many behind-the-scene efforts. The breadth and thoughtfulness of information provided by our speakers and additional discussion and clarity provided by speakers and panelists and the Q&A sessions are enormous. Thank you for your participation of day one of this workshop. I look forward to your attendance of day two of the workshop tomorrow. Day one of this workshop is now adjourned. Thank you.